Hello? I'm afraid I can't hear you yet. Don't know whether you can hear me.
Hello. Everything good? Okay, I cannot hear you. Can you hear me? Wait.
My fault, sorry.
Okay.
My fault, I didn't press the button. Okay.
Okay. Yeah, I saw the abstract, and I've little clue about AIDS or HIV, but I was curious to see how the prediction.
Right. So Leon entered, and I don't know, in the meeting, Leon here. I'm trying to press admit. Okay, joining. Finally, Gabriel is joining. Let us wait a minute until everyone who wants is. Gabriel is in process of joining. Justus and Leon here. And you are Justus, right? You are Justus Fink.
Yes.
Right?
Thanks. Yes.
Perfect. And Gabriel Chia is in the process.
Okay.
I pressed all the buttons.
Yeah.
I don't know why. Hey. Anyway, probably I will start. I don't understand why Gabriel cannot join. I pressed all the buttons. Yes,
Yeah.
right. New share. Yeah, participants are shown. Gabriel disappeared. So currently we have me and two of you. Okay, why don't I start? I think because it's already 5 minutes past. And if somebody joins, I will just let him in. Okay?
Yes, perfect.
Yeah.
Perfect. Then I am removing that. Do you hear the screen?
Yes.
Great. Do you hear models of progression of AIDS, right? Do you see my screen, right? Okay, so if anyone doesn't see my screen or my slides, please tell me, because otherwise, okay, so I would like to tell you about how I discovered what causes AIDS. Well, of course, everyone knows that HIV causes AIDS. That has been found out in 1983. This is not something which I discovered, but you see, the problem is, it doesn't work. The problem is that if you look how HIV dynamics enrolls in time, you will see that initially you have a normal CD4 count, about 1,000 cells per microliter. And then when you infect with HIV, it increases very fast rate, as shown by the red curve. And then it goes through the peaks, accompanied by mild flu symptoms. Then it dives down by many orders of magnitude. and then establishes a steady state, almost perfect steady state, plus minus fluctuations. A CD4 count accordingly is depleted by the virus, and CD4 T cell count, and then it slowly decreases. But the interesting part is that the all associated timescales are very short. Even uninfected CD4 T cells live several weeks, and therefore this decline is super slow. At every moment of time, we can speak about quasi-steady state. We have a situation of a quasi-steady state. For some reason, this quasi-state is not perfect, and eventually it results in strong depletion of CD4 T cells. And because CD4 T cells serve as helper cells for many infections we usually do not suspect, many opportunistic diseases emerge, which result in death. So it's a depletion of CD4 T cells which causes death. What is unclear is what causes this depletion, because the initial depletion is accompanied by steady state. Why steady state is imperfect is the question of this talk. If you take several models, HIV, and I investigated quite a few for the last quarter of a century, then you and many others, people, but I will show only the simplest two models. If you take simplest model, which has only replenishment of CD4 T cells from precursors, their death because of natural lifespan, and their infection by the virus. And the second equation is obviously for the infected cells, which can be infected or they die because the virus kills them. So this system has a steady state. The steady state is perfect. It means it's going to exist forever, and nothing ever will change. And that is exactly the case in sooty mangabees, in these monkeys who are natural hosts for HIV. And in them, it's a steady state forever for all, whatever, 9 or 11 years of their life, or 7, I'm not sure, but it never happens. Just there is absolutely no evidence of decline in CD4 T cell count. So it is clear that the reasons for AIDS are separated from the reason why we have steady state. It is a completely additional biological factor. So now if you take a more complex, obviously this model was too simple. In fact, we have a strong CD8 T cell response. Those are cells which are activated by virus. And well, they also have natural lifespan, and their role is to kill infected cells. So we have this additional parameter here, which was not present in the previous model. And that is the model for adults. In all adults, this is the simplest model, which is some sort of relevant here, while the previous model was from babies. It didn't include CTL response. And this model also gives perfect steady state. There is no difference. I mean, the expressions are different, of course. There are additional parameters, and actually expressions are quite different. For example, for ISS. ISS, for example, does not depend on R0. R0, I forgot to tell you, is the basic reproduction ratio, which is analogous to the number of people you infect if you go to the street without mask and whatever, bunch into people with, and you have COVID. That's the same thing. It's a number of infected cells, which cell is infects, which is a combination of those parameters, lambda, dt, k, and I. R star is another characteristic combination. So the point is that this is perfect steady state. And then, oh, now there are 4 people, very good. And then, how do we explain AIDS? I'm trying to press the button, it doesn't want to progress. Because the button is here. So where does the CD4 count decline from? The models don't predict it. And there have been a lot of efforts actually to understand that. And so far to no avail. There are 2 alternative hypotheses why in this regard. The one is the one which I'm proposing now. It is a minority view. It is not accepted at all. It's a completely novel. So you might as well hate it or criticize it because it's new hypothesis. So, and I'm going to defend it. So I'm saying that the virus is well known to evolve in HIV patient. It's extremely diverse and evolves very fast. In fact, some areas have like a 30% diversity in the virus, but mostly it's 1 or 2% diversity. If you take 2 sequences from the same patient somewhere in chronic phase, they will differ by hundreds of sites, hundreds of SNPs difference. And therefore it evolves in order to adapt to a patient. And because R0 enters all formulas, including that for TSS, CD4 steady state CD4 T cell count, well, it will decrease. R0 increases, TSS decreases, simple according to these models which I showed you. And it doesn't matter what model you take, TSS always depends on R0, and always the higher R0, the smaller TSS. The higher is fitness of virus, the smaller CD4 count in steady state. That's why we have this completely different mechanism of very small evolution with adaptation rate V, which is equivalent to the change in logarithm of R0, which causes the slow slide of the system in adiabatically slow way, as a physicist would say, if you studied thermodynamics, and towards zero. So, but the majority view, which especially I recommend you this excellent review by Louis Piquet, and it's homeostatic deregulation. That's something wrong with homeostasis mechanism. So the light brown curve is going down because homeostasis is getting worse in time. Because CD4 T cell count also proportional to that rate, it also goes with it. So it sounds reasonable hypothesis, and actually there is a lot of data which are supporting it. So I'm attacking something which is well established. I'm questioning, so don't bear me on the stage before I finish speaking. Okay. So what is the mechanism of adaptation to a host? The mechanism is that all patients during the first 6 months, as it is well known, undergo very fast selection sweeps in CTL epitopes. Not all active CTL epitopes mutate. I would say half, roughly speaking, but good portion of them are mutating very early. And it's profitable for virus to mutate on CTL epitope because otherwise it will be cleared. So
what is the function? Can you write what is the function of the CTL epitope? I have no background.
No, CTL epitope is the 8 amino acids which are recognized by a particular CD8 T cell clone.
Okay.
8, 9. So if some of these amino acids cause significant loss of recognition of binding. So I had probably to tell you about the story about how immunity works, but I don't unfortunately have time. The point is that virus, the immune cells, CD8 T cells, recognize the infected cells based on peptides exposed on the surface of cells in this in pubelle, the trash cans. Keep already forgetting English with this pair. So it's in trash cans, they expose these things, and when they bind, TCL receptor binds, and it has high specificity. It kills the cell, and also CTL replicates using that. So once you abrogate it, it's good for virus, but that unfortunately decreases replication ability. And because of that, it requires a lot of compensatory mutations elsewhere in genome to repair this damage to virus. And they appear in the order of the values of selection coefficient, of selection, fitness benefit S. So the biggest guys appear first, the smaller guy appears later, and as a result, we have hundreds of diverse sites. All of them, you have approximately 15, 20 compensatory mutation per one CTL mutation. As a result, we have 20, 25 mutations early in epitopes, and much later, after they're all done and the sweeps are fixed, you have lots of other mutations, which are compensatory mutations. And then the adaptation rate has been found for different similar models, and the earliest expression for adaptation rate has been obtained by myself in 2003, but now we have a much better expression for distributed S. But this is also well doable. Basically, it is expressed in terms of number of infected cells, selection coefficient, mutation rate per genome. It's a very simple, compact expression. And the dependence is logarithmic on everything, basically, except for selection coefficient. Please note that selection coefficient can be measured in patients using the methods which developed by me, by Richard Nair, and yesterday and Friday I heard excellent talk. I think it was Barton. So, and it's still another method of measuring selection. There are plenty of methods of measuring selection coefficient in patients. And that's very important because it means that we can predict adaptation rate just by looking at a slice of genomic data. Now, if you have recombination, the expression is different. It's as is replaced with R. But that applies to high viremia patients in whom recombination is important. Low or average viremia patient, recombination is not important. As a result, we can predict time to AIDS as a simply, obviously, the difference between logarithm in TSS divided by the derivative of the logarithm. And derivative of the logarithm, because it's inversely proportional to R0, is the same as V, except for the sign, right? So, so this connects to this log derivative, and this gives you V. So immediately plugging it in, you get the simple answer in the absence of immune response. As simple as that. This is normal count. This is say 1,000. Maybe it's actually more like 500 because it's already after infection steady state. I apologize. This is 500, not 1,000. And typical patient. And this is 200. So logarithm 2.5 psi is very close to 1. So TH equals 1 over V. Simple. That's a prediction. And yes, it gives you 1 or 2 years. It gives you 1 or 2 years, and that's exactly what we observe in babies. AIDS occurs in babies who do not have immune response in 1 or 2 years. If we include immune response, which is necessary for adults, TSS is delayed, and we obtain somewhat longer formula and longer time to age. And again, psi is the same as about 1. And as a result, we have 5 to 10 years prediction in an average patient. This is exactly what we observe. So, okay, great. So this model at least gives the correct ages for infants and adults. How about the correlations? It's well known that time to AIDS inversely correlates with virus load, as shown here. Correlation is pretty slow. It looks like something like a square root or logarithmic correlation. It's hard to tell. And the red lines show 2 parameter fit of this formula. So in fact, it has 3 parameters, but one of these parameters you can like C keep constant and use only A and B to fit. And that's the result of fitting. Furthermore, actually you can fit with a single parameter because if you change B, you can compensate it by a change in A. It's 1 parameter fitting. So this formula is so well describes this that you can use 1 parameter in order to fit it and not 3 as it appears here. So therefore, okay, so it's good. We can explain the observed inverse correlation between time to AIDS and virus load in a particular patient. I hope I explained those are data, each dot corresponds to 1 person who is infected with HIV, is not treated with drugs. I don't have data on treated people, unfortunately, but in principle, this formula should apply to them as well. Okay, the alternative is a limited division model. It basically says, the simplest realization of this model says that, first of all, we have to remember that replenishment rate is not constant. It has a logistic term 1 minus T or T0, which turns off replenishment when T reaches normal level. That's now we have to be more accurate with the replenishment rate of T cells, now which are infected. This model postulates, if you read this review, that the number of divisions of the precursors of T cells is limited because of telomeres. This is assumption. In this case, it is expressed in the fact that you can produce only finite number of precursors. Lambda T times T8 equals lambda. Once this limit is reached, that's the time to AIDS. As simple as that. We run out of cells. A simple model. It's not obvious it's correct because immune cells produce telomerase protein, which restores telomeres back. This little tail at the end of DNA is being restored. And this model predicts abrupt drop in CD4 T cell count and time. Unfortunately, it is not observed. When you run out of cells, you have a drop. And unfortunately, it predicts this green line, which is very curved. And I don't know, you cannot really say that it contradicts to this data because data have a lot of dispersion, but it's definitely, there is nothing in data which would hint that you have this bend. I would say that the fitting is not as quality. Perhaps I should provide you the p-values, and my reviewers actually of the paper asked me to do it, but I don't have p-value at you yet. So, yes.
Can I ask you?
Sure.
So this is data, each point is a different patient, right?
Yes.
So is there a series? No, it has to be different patients.
Somebody is asking to admit. I have to, sorry, to disconnect. I'm trying to do it, but why are they asking to admit? I think it should be automatic on admission, no? Okay, I'm sorry. I'm always very nervous.
No, go on. No.
Okay, somebody wanted to admit. No, okay. Here, chat. No, okay. Anyway, I didn't have time to press the button. Let us do again. So, yes, each point is one patient. Next.
Okay, so can you also compare to dynamics within patient? Basically, if you look at the decline of the counts within a single patient over time, and then
yes, I can. I don't do it in this talk. Such data are available. It's there is strong fluctuations, but generally, it's if you average out over many patients with a linear decline. Basically, it's with very strong fluctuations. Some of them are like concave, some of them like you know, some of them the other way. But if you average over patient, on average, it's a linear decline in time, just linear. Boring linear decline as shown in the first slide, nothing else. That model, the second model, assumes that it's erosion of the source by virus. So the idea is that the virus somehow accumulates its products, damages this environment in lymphoid tissue, so that, you know, there is less and less replenishment because of virus products. And that is nicely expressed by this simple formula, which says basically that the source decays exponentially proportional to virus. In this case, time to AIDS is simply when TSS reaches the level 200, which is supposed to be for AIDS, 200 cells per microliter, which is where the symptoms emerge. Some people say it's 350, but typically I think CDC still says 200. So that's what I used. So once you do that, you obtain this formula for AIDS, where this is a source before decay, and alpha is somewhat this term, this coefficient. And if you call this beta, then you have again two parameter fitting. So everything is again two parameter fitting. And if you alpha and beta are composite parameters, so if you fit, you get the blue curve. And it's way too sharp because it's 1 over alpha. It's well known that this can be a logarithm or a square, but it cannot be just 1 over virus. It was known for a long time that this is not 1 over X correlation. So this model is simply terrible, and there is beyond doubt it's not suitable. We can talk about the green, but green, as you said, as I pointed out, predicts abrupt drop. I have never seen abrupt drop. There are all sorts of curves between different patients. They vary like crazy, but on average they are linear, and I never saw abrupt drop. So I don't think it's really. So virus adaptation model predicts decline in CD4 T cell towards AIDS. Predicted range of time in infants and adults is correct. It fits the form of negative correlation between time till AIDS with two parameter fitting. And it's much better than the homeostasis model, especially this model with, which assumes decay of homeostatic environment by virus. And most importantly, it can be used as a prognostic tool because we have so many methods of estimating selection coefficient from sequences in a patient. And so we can easily, oops, Balok entered the room, I think it's next, that we can basically very easily predict this. And you can directly measure the number of infected cells by the proxy. So now I can take a blood sample from a patient and measure S, sequence him, estimate S, measure his viremia, and then I can tell when he's going to have AIDS. And that applies to treated and untreated patients equally. Thank you. And
cool.
One thing, am I over time? Am I over time? I'm not over time yet. Great. So I would like you to, everyone must buy my books, the sales pitch. No, it's, I just wrote a book. I'm very proud of it. Okay. So, and it's very mathematical. doesn't have any data in it at all. It's just pure math about the principles of evolution for those 80 years. So, and it's great as a textbook for a person who have a graduate level of, who is from STEM sciences especially, and who is interested in learning about evolution. Okay.
Cool. Thank you.
Okay. Any other?
Thanks.
Yes. Can I ask about the puzzle with the monkeys? So the monkeys, they don't have this slow declining from this steady state kind of.
Sutimangabis, specifically Sutimangabis.
Okay.
There are actually, if you take a strain which is native to the monkey, you will not have a decline, yes. If you take, for example, SIV 239, 251, and put it in macaques, you will, rhesus macaques, you will have a decline. And this strain was specifically found to imitate pathogenesis of HIV in humans. So it depends on whether strain is in its natural host or not.
Okay, because I wonder if you can use this to kind of distinguish between the different hypotheses, between the homeostatic deregulation and the viral evolution. Because if it's a property of the strain, as I just understood, right, then it should be.
Yes, you're absolutely correct. This speaks in the favor of evolutionary model because, I mean, the other stuff is everywhere. The homeostatic equilibrium the homeostatic argument doesn't depend on the strain, it doesn't depend on the host, it should be very universal. And while here, that's the fact that if you have a match of virus to its natural host, you don't have AIDS. That's correct, because it's already evolved. And in humans, of course, we are not evolved. It's only, 70 years after entry to humans, right? So no, it's not evolved. Do you still see the slide? So it's not evolved yet. And first of all, it's not evolved to humans as a species. And second of all, it doesn't evolve to humans as individuals, because location of CTL epitopes in each individual is varies, depending on your 6 of 6 HLA subtypes. So therefore, because the mutations location of CTL epitopes is completely like random, if you change between you and me, maybe we'll have some coincident HLA subtypes because 6,000 years ago, whatever, our ancestors came from the same place in Ukraine, but generally between two individuals, it will be quite different. And that causes also the variation of the variation of different diverse sites. So yes, you're correct. If virus evolved, now imagine that all most important HLA subtypes already, suppose virus already mutated in all HLA subtypes for the given species of monkeys. It's already pre-mutated, like millions of years ago. So it doesn't have to do all of that. It's already there. It's already at the peak of it. And then, of course, because these monkeys say share similar HLA subtypes. So at all those things, it's already pre-mutated. And then we have a very minimal evolution. And that also explains inverse correlation with immune response, because it's known well that the higher is immune activation in humans, the faster there is progression to AIDS. I didn't discuss it. The same thing. It's well, okay. Because it's CTL response which causes evolution in the first place. So if you have if a response is less active, then you have less evolution, and you have the progression to AIDS slows down. So the model indeed explains pretty much all facts naturally, and as you can see, equations are super simple. So okay.
Yeah, I think it's cool to have like this old puzzle and the simple model.
Yeah, I'm very excited. And I forgot, by the way, mention about these monkeys. Thank you for reminding me, because I just received today request for revision of this manuscript, and I forget to say about monkeys. Thank you.
Okay, I think Leon wanted to ask too, right?
Sure, Leon.
That's fine. That's fine. I think we should move to the next talks.
Sure. Excellent.
Nice to see you, Leon. Hi.
Hi.
Okay, let's move to the next talks. Now, what I do, I have to mute myself, right? And that's it.
So who's next? I mean, how does it work?
I think we have to move to another Zoom room.
Okay.
This is another Zooming room, right?
Yeah.
Okay, then
So
everything, thank you. And let us, even if it's the same, we will see it. So don't worry. So thank you very much and goodbye.
bye.
Bye-bye.